Imagine you have a million of these cells where you cram a bunch of DNA into it. And you do this a million times. Two parts of that DNA molecule, two parts of that spaghetti noodle, if they're close together linearly, they're going to be more likely to touch each other in three-dimensional space. Sequences that are close together on the chromosome are more likely to be close together in three-dimensional space. And now you have thousands of millions of cells of the same organism, blood, sample, whatever. And you're measuring how much touching there is between everywhere in the genome. If you average out that signal across all these cells, what you're going to find is if things are close together, they touch more. If things are further apart, they touch less. So you're saying if this touches more, then it's close. If this touches less, then it's far. We have figured out how far and how close every part of the genome is on the linear scale. And back in those days, this is about 10 years ago, you could sequence a genome. You could read all the letters of the genome. The hard part was putting them together. So imagine you take a book, you shred it into little pieces, and you copy those pieces 1,000 times. You go, I just sequenced this genome 1,000 times, 1,000x. And that is true. Like you have a jumble of sequence. It's not the genome. It's just the contents of the letters of the genome. So actually putting it together is much harder than sequencing it. And so you try to take little pieces, stitch them together into longer pieces. But eventually what you're trying to do is get the whole genome, the whole chromosome. And the process of putting little pieces into big like chromosomes is called scaffolding, right? Kind of like how you like scaffolding is you're making these scaffolds where you put the pieces into place. It's a bit like putting jigsaw puzzle pieces into the puzzle.